When you eat a meal, you probably don't think much about what happens next. But what if I told you that your food is about to go on an absolutely wild chemical journey? We're talking about a trip through some truly extreme environments, and it's all happening inside of you. So let's pull back the curtain on this incredible internal chemistry.
And it all really boils down to this one central puzzle. How in the world can your body break down food first in a solution that is acidic as, say, battery acid, and then, just moments later, finish the job in an environment that's almost totally neutral? I mean, it sounds like it should be impossible, right? But our bodies pull off this amazing feat with every single bite we take. And this really shows you the tale of two completely different worlds we're talking about. First up, your food hits the stomach, which has a pH of about 1.5. That is incredibly acidic. Then it moves on to the small intestine, and whoa, the pH jumps all the way up to a really gentle 6.5. So the big question is, how does anything manage to get digested across such a massive chemical shift? Well, the answer lies with these little unsung heroes of our biology, tiny, microscopic machines called enzymes. They are the absolute key that makes this whole incredible process work. They're the stars of our show today.
So what are they? To put it simply, enzymes are just proteins that act as biological catalysts. They speed up chemical reactions. And for digestion, we're talking about a specific type called hydrolytic enzymes. Their whole job is to chop up all the big, complex molecules from your food into tiny little pieces that your body can actually absorb and use. Okay, so how do they actually work? Well, every single enzyme has this special little nook called an active site. And you can think of it like a perfectly shaped lock. It has a super specific three-dimensional shape that only one specific key, its target molecule, can fit into. And this perfect fit, this lock and key mechanism, is absolutely crucial for the enzyme to do its job. But there's always a catch. And here it is. Just like any highly trained specialist, enzymes are, well, they're picky. They are super picky about their work environment. They can't just operate anywhere, under any conditions. They need things to be just right, and that is especially true when it comes to pH. And this perfect condition has a name. Scientists call it the optimal pH. It's that one specific level of acidity or alkalinity where a particular enzyme is basically at the peak of its powers. It's working faster and more efficiently than at any other pH. This is its happy place. And you can see it perfectly right here. If the environment gets too acidic, look, activity drops right off. If it gets too basic, same thing, activity just plummets. It's only in that just right Goldilocks zone, that optimal pH, where the enzyme is really firing on all cylinders. But why? Why does this happen? What is it about the wrong pH that just completely shuts an enzyme down? To really get that, we've got to zoom in, way in, all the way down to the molecular level. The whole secret? It's all about electricity. See, an enzyme is a protein, and it's folded into this very, very precise 3D shape. And that shape is held together by all these little electrical attractions between different parts of the molecule. When you change the pH, you start messing with those all-important electrical charges. Let's get a little more specific. Enzymes are made of amino acids, and these have important chemical parts, like amino groups and carboxylic groups. Now look at this table. In the superacidic stomach, that carboxylic group stays neutral, no charge. But once it gets to the neutral small intestine, bam, it suddenly becomes negatively charged. That one tiny change in its electrical charge completely rewrites the rules holding the enzyme in its proper shape.
It really is a powerful domino effect. It starts when the pH of the solution changes. That makes these key chemical groups either gain or lose a little hydrogen particle. Then their electrical charge gets altered. That change disrupts the super delicate folding of the protein, which then warps the shape of the active site. And if the lock changes shape, well, the key doesn't fit anymore. The enzyme is basically switched off.
And the source material really puts it perfectly. It says that when the protein's folding is altered at the active site, the enzyme just can't bind to its target anymore. The connection is broken. Game over for that enzyme. Okay, so now that we've got the science down, let's circle all the way back to our meal, making that big journey from the stomach into the intestine. How does our body actually solve this huge pH problem?
Well, the body's solution is it's so clever. As that acidic food mixture is leaving the stomach, the pancreas squirts a buffer, bicarbonate, which is basically just baking soda, right into the small intestine.
And the effect of this is instantaneous and, honestly, pretty dramatic. It totally neutralizes that powerful stomach acid, causing the pH to just skyrocket from that harsh 1.5 all the way up to a much friendlier 6.5.
And here is the truly brilliant part. In that moment, the stomach enzymes, which love the acid, are instantly switched off. But this new, neutral environment is the perfect sweet spot for a whole new team of intestinal enzymes to jump in and take over. It's like a perfectly timed, perfectly choreographed biological handoff.
So, let's just quickly recap this amazing process. What are the key takeaways? First, enzymes are specialists, and they need a very specific pH sweet spot to work. Second, if that pH is wrong, their shape changes, and that deactivates them. And finally, our bodies are super smart. They use buffers like bicarbonate to create these different pH zones, allowing different teams of enzymes to do their jobs in perfect sequence.
It is a stunningly precise chemical ballet, and it's performed flawlessly with every single meal you eat. It kind of makes you wonder, doesn't it? What other incredible, invisible wonders are happening inside of us, right at this very second?